Welcome to today's webinar about healing the leaky pipe, esophageal barrier dysfunction in EoE, featuring Dr. Joanne Masterson from Maynooth University. My name is Jennifer Roeder. I'm a communications consultant for APFED, and I'll be moderating today's webinar. We are pleased to have Dr. Masterson join us today to share information about translational research. Dr. Masterson's expertise is in the area of epithelial and stem cell biology, and she has spent nearly 2 decades researching eosinophilic gastrointestinal diseases. She's currently the group leader of the Allergy, Inflammation, and Remodeling Research Lab at Maynooth University, also known as AIR. Her research laboratory investigates the impact of allergic inflammation on the function of the esophageal epithelial barrier and associated stem cell niche. This work is focused on finding new and innovative ways to treat patients with EGIDs, with a particular focus on eosinophilic esophagitis, or EoE. Welcome, Dr. Masterson.
Great. Thank you very much for having me, and thank you very much for the invite. And for those of you attending, thank you very much for your interest in our presentation today. So, as nicely introduced there by Jennifer, I'll be talking to you today about healing that leaky pipe. So what's happening with the esophageal barrier in patients with EoE. And as Jennifer introduced, I'm a group leader here at Maynooth University in Ireland. I previously spent 10 years working with the GEDP program at the University of Colorado and the Children's Hospital Colorado. So what do you think when you hear a leaky pipe? So you probably think something like this, chaos. So we're not really proposing that this is the kind of thing that's going on with patients with EoE, but something more like this, a slow drip that's on and off over time, something that can cause a problem though if you don't tend to it. So as I put this presentation together, I wondered whether that makes me a scientific plumber, but I guess maybe you can tell me at the end of the presentation whether you think that's the case. So the goals and objectives of today's presentation, what will we learn today? So I hope to introduce you to what translational research is. So fire some of those brain cells, light up some of those light bulbs. And tell you how we know that the pipe is leaky, as I described it in the title, and to tell you a little bit about what translational tools we have to be able to study this leaky pipe. And at the very end, in a short few slides, I'll give you some information about my research specifically, what I found and what I'm continuing to examine in this process. And in all of this presentation, I hope not to blow your mind. So what is translational research? So it's the rendering of something by definition into another language or into one's own from another language. So it is the translation from something you don't understand to something that you do. But from a research stand and from a scientific point of view, that's slightly different. So translational research by definition takes scientific discoveries that we've made in the laboratory, or those that we've made in the clinic or out in the field. And it transforms them into new treatments and approaches to medical care that improve the health of the population. And although these are fantastic definitions, they're very linear in their description. And I think of it more as a dynamic process, translational research. So to me, translational research is very well epitomized by this diagram from the NCATS, the National Center for Advancing Translational Sciences at the NIH. And it very much encounters or encompasses what I would consider translational research to be. So my research is over here on the right-hand side. It's the basic research, it's the preclinical research. And I'm very much informed by my colleagues at Clinical Research Encounters, their clinical implementation, and of course, our understanding of public health. And most importantly, I'm informed by the patient's involvement and the patient's needs. And together, this is what I consider to be translational research. And this is where my research sits within this framework. So moving on then, how do we know that the pipe, the esophagus, is leaky in patients with EoE? And there have been extensive investigations to examine this. But in 1992, this is what we knew about eosinophilic esophagitis. We knew nothing. It was a brand new need that we didn't know was coming around the corner. And in 1992 and in 1993, a number of publications of a cluster of patients were identified in Switzerland and in the US, where a preponderance of eosinophils within the esophagus was described of these patients, a curious gathering of eosinophils. And this describes to you the area of significant growth that has been the research field looking at eosinophilic esophagitis. So if I can just walk you through the slide very slowly, over here on the y-axis, we have the number of scientific and clinical publications per year. So we can see over time, working from 1993, those very first publications, where it wasn't yet defined as a disease, but was being observed by patients or doctors in clinics, through till present day, till 2021. In the red bar, we can see here a dramatic increase, a rapid increase in the numbers of publications, and therefore a rapid increase of our clinical and basic science understanding of this disease. And just for comparison, I have here in the orange bar, publications pertaining to allergy and eosinophils. So why is this important for me to share with you? So those original discoveries or those original publications of the patients who had these eosinophils by our clinical colleagues were published in 1993 by Stephen Atwood and Alex Straumann in 1994. But it took till 2007 for the first clinical consensus diagnostic guidelines to be published by Glenn Ferruta and the team. And in 2011 and 2018, these were updated. But from my perspective, here's the crazy little scientist in the lab. I finished my PhD in 2007. I was emerging into the field of research, trying to find what I might focus on as I moved my research career forward. And coincidence brought me together with Glenn Ferruta and the field of EoE in 2008 to begin my postdoctoral research. So I came in just as this graph was taking off. And a huge part of this work has been supported by people like APFED through the years in trying to advance our understanding of EoE. So what do we know now because of all of those clinical and scientific publications? We know that the age of onset for EoE is bimodal. So there are two key times where EoE can often be diagnosed. So in the youth, in children, and in adults. So it is a disease that is chronic and is found in both children and adults. We know now that it's part of the atopic march, that this allergic disease, eosinophilic esophagitis, can be found in patients who have other allergic diseases such as asthma, food allergies, atopic dermatitis. In this review in the New England Journal of Medicine in 2015 by Glenn Ferruta and David Kotska, they showed that patients were found or summarized studies where it was assessed in patients, the genetics, and there was found that there was some uniqueness to the genetics of some patients who had eosinophilic esophagitis. But we knew that in the context of these patients, if they swallowed a food antigen or an aeroallergen, or sometimes an environmental factor through the esophagus, this led to an inflammation described over here in this box, and that inflammation presented itself as this eosinophilic esophagitis, this endoscopic picture that you can see here was very abnormal, these white exudates, these plaques, these gatherings of eosinophils on top of the esophagus. And this esophagus here should be smooth and bright and translucent, and you can see it's not. It's very inundulated with these linear furrows, these concentric rings. And the chronicity of that inflammation, unfortunately, will lead in some patients to fibrosis and dysmotility, to food impactions, to dysphagia, difficulty in swallowing. So the symptoms of patients with eosinophilic esophagitis change as patients have the disease for longer or in the age of the patient presentation. So developing from a feeding disorder or vomiting in the very young children to abdominal pain and difficulty swallowing or dysphagia here in pre-adolescence, adolescence. And then in our teenage years, we see an increase in food impaction as a symptom in patients with EoE. And more recent work by the Consortium for Eosinophilic GI Researchers, the SEGER Consortia, found very nicely when they combined a number of different factors. So they looked at the onset of the disease, whether patients had an atopic status, the endoscopic phenotype, so looking down the camera in that endoscopy and quantifying that using a numerical code, as well as the patient's response to topical steroids and a molecular phenotype, they were able to describe that there were three clusters of patients with eosinophilic esophagitis, and even suggested that potentially these clusters are a continuum from one through to the other. So we've had significant advances in very many ways in understanding this disease from a clinical perspective over the last 2 decades. The current treatments tend to be the 3 Ds, so drugs, diet, and dilations, but there's still a small portion of patients who are non-responsive to these therapies. And we also know that trials targeting a number of different factors have unfortunately been inconclusive to date, although there are a number of promising clinical trials ongoing and in the pipeline. And we also know that this disease has a significant impairment on the quality of life of the patients who live with it and their families. So going back to this review of what we know to date, to highlight for you in the center here, this barrier function that has been highlighted to be particularly important in patients, in the pathology of patients who have eosinophilic esophagitis. And this barrier dysfunction is the focus of my research in our research group. So what causes eosinophilic esophagitis? So we know that it's an allergic reaction within the esophagus. We know that we swallow airborne allergens or food allergens, and these lead to an inflammation within the esophagus that involves a number of different inflammatory mediators, and these inflammatory mediators elicit a damaging response within the epithelium that leads to the symptoms that these patients experience. So since 1992, there's been a rapidly expanding and exciting expansion of the clinical tool belt, both in the diagnosis of eosinophilic esophagitis as well as in therapies. And we can see here the endoscopy EREFs, so the scoring system for that endoscopy that I mentioned in the previous slide. We've also developed or advanced our understanding of allergy testing in patients with EoE, the use of radiography to look at that narrowing of the esophagus from the chronicity of inflammation, and critical to our understanding of patients with EoE, the pathologist. So they've developed a very elegant and informative histology scoring tool that we were able to use in the standard of care for patients. A number of other scoring systems have since been developed and validated in patients with EoE. But more on the investigational side of this clinical tool belt are things like the functional luminal imaging probe, so the FLIP device or the impedance devices. And these can look at dysfunction, literally, of the esophagus. And we'll talk about them in a few slides to come, as well as molecular profiling of patients with EoE. So how have we studied barrier in patients with EoE? How do we know that pipe is leaky, as I said? So the first piece of evidence that I put forward to you is genetic, the genetic underpinning, that it's written in our DNA. And if any of you have ever been involved in clinical research, you understand that sometimes you might donate to these genetic studies where a vial of your blood might be taken. And in that blood, we're able to isolate the DNA from your cells that identify you as an individual. And that DNA can then be analyzed in the lab, and we can look for differences between you and the general population or a control population we're comparing to. So in these early GWAS studies, a number of different genes associated with barrier were found to be different in patients with EoE. And one of these genes, calpain-14, identified here on the top left, was taken into a translational study. So this finding in the clinic was brought back to the lab. And what they found is when they mutated this calpain-14 protein, in comparison to this normal cell culture here on the left, this is grossly abnormal abnormal, indicating that there is a deleterious effect of the abnormality in this calpain-14 protein that was identified by genetic studies underpinning patients with EoE. Another way of analyzing barrier in EoE is looking at gene expression. So I'm going to put on my lecturing hat here, my science teacher hat, and I'm going to tell you that there's a central dogma in molecular biology. And the central dogma goes that from the left here, your DNA makes a messenger called an RNA. That RNA then is made into a protein, and that protein is what tells our cells what to do and how to do them. It makes them function the way they should. But that probably makes as much sense to you as this disorganized picture of somebody's kitchen down on the left. So let me translate that a little bit, hopefully into something you might associate with better. Our DNA is the equivalent of a cookbook. And that cookbook wants to make some tasty pastries that fill our bellies. But in order for those pastries to be made, we need the instructions. And the instructions are this RNA molecule that's in the middle. This translation or transcription product, this RNA. So the RNA can be assessed in a methodology called the transcriptome. And the transcriptome is basically the recipe or instructions that we use to make a cake. It's the recipe or the instructions that our cells use to make proteins, to make our cells work the way they should. And in the focus of this talk, to make proteins that are important for barrier between the food that we ingest, the air allergens that we swallow, and the immune system that underlies that epithelium in the esophagus. And we're going to ask the questions in our research. Did those instructions get mixed up? And most importantly, can we translate it? Can we rearrange those instructions? Can we fix them so the cake can still be made? So in number of very elegant transcriptomic studies in the very early days show that there are a number of genes that are altered in patients with EoE. And a lot of these genes cluster on chromosome 1. And so you can see here, in comparison to the normal individuals that were enrolled in this study here on the left, patients who were suffering from eosinophilic esophagitis had a significant decrease in a number of these proteins, or the number of these RNAs, these messengers, had been decreased in patients with EoE. So the instructions had gotten lost. They were mixed up. And when they looked more specifically at these instructions, they found that in the blue bar here, you can see there are a number of those instructions that are switched off, that are down in patients with EoE, compared to the numbers that were up. And when they took these genes, these instructions, and they put them into computers and asked the computer to tell them what those genes, what those RNAs might be involved in, the biological processes that they were involved in were very much associated with barrier. They were involved with keratinization, keratinocyte differentiation, epidermal cell differentiation, and so on. So these were all involved in those processes that were downregulated in the instructions manual for patients with EoE. And these processes that I've mentioned there are indicated here in a pictographic way. So on the left here, we have a control patient's esophagus biopsy. And you can see here the very normal architecture. So these dark blue cells are stem cells, our progenitor cells, are cells that then give rise to daughter cells. And those daughter cells sit here in the middle. And then those daughter cells change their function, change their protein. And that protein in these cells is what gives this epithelium its barrier. So this is our lumen out here. This is where our food passes through. And under here is where our immune system would normally lie. So this is a very normal looking esophagus where we've got a green light from bottom to top. But in a patient with EoE, we see that that's much different. So we can see that it's a more patchy, irregular progress of cells from the bottom to the top. And this process, these biological processes, are what are switched off. And they're the ones that are important for pushing those cells up through this process. So you can see here a genetic signature of a analysis here. So this is likely a control patient grouping here, and this is likely a cluster of patients with eosinophilic esophagitis, showing differences in gene expression in these patients. So other evidence of barrier dysfunction is that of ultrastructural features. So what's ultrastructural? So let's think about what we can see with the naked eye described here in this graph and in numbers of size. So as molecules or things become smaller, so our cells are much smaller, we need a microscope and a camera to be able to see those things. But in order to see molecules within the cells as small as an atom, we need a microscope called an electron microscope. And this is a very large piece of equipment here described on the left. So using an electron microscope to get right down to the atomic detail of tissues from patients with eosinophilic esophagitis, a number of very elegant studies, including our own, looked at cells and tissues from patients with EoE. And what we found, this is a cell here from a patient with EoE. And these dark patches that are highlighted with the yellow arrow are connections between that cell and the cell that's adjacent to them. And what we can see in this patient who has eosinophilic esophagitis, there's a lack of these yellow arrows. There's a lack of those electron dense aggregations or connections between the cells. They're not sticking to each other the way they should. And we were able to count that and compare. So compared to a patient or a normal individual with no eosinophilic esophagitis, there was a decrease in these cellular connections in these patients with active EoE. And excitingly, when patients were treated and there was a resolution of the numbers of eosinophils, so they were found to be inactive, there was a restoration of that barrier function in those patients. And other studies have shown something similar. So looking at the space, this white space between the cells, looking at how big that is, we could see that in this study, compared to control subjects who had enrolled in the study, patients who were experiencing a diagnosis of EoE had much greater space in between their cells. So this barrier, this pulling together of cells, the knitting together of that barrier isn't occurring properly in patients with eosinophilic esophagitis. And that was shown that in this study that before treatment, there was an increase in that space, but that after treatment, that space could be reduced back to normal. So the last of the evidence to show that barrier is dysfunction, the pipe is leaking in patients with EoE, is electrophysiological assessment. Big words for me. So you might be familiar with something like this. This is an EKG, if my clinical colleagues would inform me, but this is a measurement of heart function. So essentially we can do the same to epithelial cells, but look at barrier function. So just look at the next graph and think the bigger the number, the better the barrier. And in this study here, they showed that in patients at baseline here in the black bar, compared to control healthy individuals, the barrier number, this tier number, is significantly lower. In patients who were treated but were non-responsive to that treatment, that barrier remained low. But when patients were successfully treated, so they were responsive to their treatment, there was an increase in that barrier, getting closer and closer to that of control individuals with no inflammation. And in another study here, again, remember, the bigger the number, the better the barrier. So in a control population here, there's a nice high level of barrier. But here on the right, patients with EoE have a attenuated barrier, a decrease in that barrier. So electrophysiologically, there was evidence of barrier dysfunction. And the last is histological. So taking a microscope, putting the sample under the microscope and looking through a camera at a computer, what's happening to the barrier in patients' specimens with EoE. So you can see here, hopefully, these halos that are surrounding these epithelial cells, indicated by these long, narrow arrows here. That's that dilated space that we're talking about. And you can see it very clearly here in this histological measure of dilated spaces in patients with EoE. So sadly, the pipe is leaking. But what translational tools do we have to study these leaky pipes? So what's the investigational tool belt for preclinical studies? So for this, we've expanded since 1992. In 1992, we had nothing. In 2008, when I started, there was very little. But we've developed a number of models to understand this disease. So we've developed animal models, we've developed cell culture models. So models that don't always necessarily need to involve the patient outright to start with, but always need to come back to the patient to better understand the relevance of those studies. So these studies involve mouse models. They can involve cell culture systems that try to mimic what's going on in the esophagus, but then taking these back to the translational relevance in biobanks from specimens taken during research with patients, but also looking at muscle contractility. So we can take the animal models and look at contractility of the muscles. We can't unfortunately do that in patients, as well as imaging. So we can do some very complex imaging with some very interesting molecules that can tell us what's going on in the esophagus that unfortunately we can't give to patients, but we can give them to mice. So we can use these mouse models to study these things. So how do we interrogate EoE? So here on the left is a picture of a human esophagus. And what you see on the top here in this white box, that's the mucosa. That's the epithelium that I study. And that's the biopsy. That's the area that you would normally take in an endoscopic biopsy in the clinic, in the hospital, if you were ever undergoing an endoscopy. So the area that we study, or we can study by taking a biopsy in the clinic, is much less. It's 0.01% of the surface area of an esophagus. If you compare that underneath this, there's a rich complexity of tissue. There is a rich complexity of muscle that helps with peristalsis, the movement of our food down through the esophagus, as well as the immune system. So this is a collection of immune cells here that underlie that. And in these biopsies, we're not able to study that underlying tissue. So we need something, we need an animal model to be able to study deeper into the tissue, as well as the epithelium. So we set about developing an animal model, and for simplicity's sake, let's just call this MIL5. This is a goodie, a food that eosinophils love to eat, and they're very happy and they live very long and happy and productive lives if they've got lots of IL-5 around them. So we took this IL-5 molecule and we overmade it. We made it overexpressed in the esophagus of mice. So we made a mouse that made too much of the eosinophil goodie in the esophagus. And then we took that mouse and we educated its immune system to a chemical irritant called oxazolone, or OXA for short. So its immune system knew that this chemical it didn't enjoy, it didn't like, it would mimic the idea of your own immune system being sensitized to a food allergen or to an environmental allergen. We then took those mice 5 days later and we dripped this chemical irritant into the esophagus on days 5, on day 8, and day 12. And this chemical irritant then led to an increase in the numbers of eosinophils that were present in the esophagus. So for those of you not familiar with the mouse esophagus, this is the lumen. This is where our food, the mouse's food, will pass through. And this is the underlying immune system here in the lamina propria. This is the muscle that we cannot normally see in patient biopsies. And here we have that epithelium where the mucosal biopsy would normally be taken from. And in the red staining here, these are eosinophils that are packing themselves into this esophagus from this mouse that has been challenged the way I previously described. And the normal esophagus would not have any of these eosinophils, so it would just be completely blue. We can also see that in these mice, we have these aggregations or these microabscesses of eosinophils, these collections of eosinophils on the surface. And that's the equivalent to those white plaques that I described in the endoscopic picture earlier on. So we were very excited because we now had a mouse model that at least histologically looked very similar to the histological picture that we had seen in patients with EoE. Even further to our excitement, we were able to show that this mouse model developed fibrosis similar to patients with EoE. So this, again, this is a biopsy that would be taken from a patient. Up here is where the food would pass through. Under here in this blue area would be where that immune system lies. But all of this blue staining is a patient who has experienced fibrosis, a stiffening of the esophagus, that stiffening that can potentially contribute to the difficulty swallowing and the symptoms that patients often describe with EoE. And what we found here in our animal model in this blue staining, again, just to orient you, up here is the lumen where the food passed through, here's where the immune system lies, and we can see a great increase in the amount of this blue staining that is very similar to what we had seen in patients with EoE. So we're very excited about the potential of using this mouse model to study mechanisms and novel therapeutics for their potential in EoE. So we need to first ask the question, though, could this mouse model be treated by existing therapies for EoE? So on the left here, we have an esophagitis mouse. This mouse is mimicking EoE, eosinophilic esophagitis. Here are the red cells, those eosinophils in the esophagus of this mouse. But what you can see here in the middle is a mouse that had been treated with dexamethasone. So dexamethasone is a steroid, a glucocorticoid, much like a lot of patients with eosinophilic esophagitis are treated with. And what you can hopefully see is a very normal mouse esophagus and a complete absence of the eosinophils now in this mouse. So we were able to quantify the decrease in both the rearrangement of the esophagus, the epithelial histology score being improved significantly with steroids, and also the numbers of eosinophils here on the bottom. So we completely depleted the numbers of eosinophils using steroids, as we might have expected. But now we can use this to test new or investigational therapies. So here again in the middle, this is our positive control. These are mice that are experiencing eosinophilic esophagitis. You can see these pink cells, hopefully, that I'm pointing out or circling. These are eosinophils aggregating within the esophagus of this mouse. In the middle, we have this stain called Chi67. So this brown staining that you see here, you can see an increase in this mouse in the middle compared to a control mouse. And what this mimics is the basal cell hyperplasia that we often describe in the histology scoring systems for patients with EoE. So this mouse has that basal cell hyperplasia, and that's further validated by this marker of basal cell hyperplasia. So this P63, this would be a very normal localization or number of those P63 positive cells. And you can see that that's greatly increased in our mouse EoE model. But when the mouse was treated with an investigational compound, we'll call it compound X, we see a restoration and a reduction of those basal cell hyperplasia indices in the mouse model. We were also able to quantify the numbers of eosinophils. So comparing to control mice, compound X treated mice had a significant reduction in the numbers of eosinophils that we were able to count. So this model system can now be used to preclinically test drugs and pathways to try and come up with new therapeutic options for patients with EoE. So in the last part of the presentation, I just very briefly, in a couple of slides, want to go through some of my research, what I found and what's ongoing. So to study barrier in EoE, we have a number of model systems that we can use for cells and culture. And this is a cell culture incubate. This is one of our investigators, Sinead, our postdoctoral scientist, embarking on her career in eosinophilic esophagitis this year in the group. And you can see all of these flasks that are stacked up. These are cells that we're growing in the incubator, trying to study eosinophilic esophagitis. So we can take these cells and we can put them into a little dish. And these little dishes, if you think of them as a casserole dish, and you've got your epithelial cells in there. So they're cooking away in their soup, happy enough. Then after a couple of days, we reduce the amount of soup on top of the cells. So the cells are now sitting in between the media that they're fed through and the air on the surface. And this causes the cells to grow on top of each other, very much mimicking the epithelium in the esophagus. So now we can study this artificial in vitro cell culture called air-liquid interface. And we can study function, barrier function. So using electrical impedance, like I described in patients, we can look at the conductance of electricity across this epithelium. And obviously, as I said earlier, the bigger the number, the better the barrier. So if we see changes in that, we're able to study in the presence and absence of inflammatory mediators, in the presence and absence of novel investigational drugs. And similarly, we can study permeability. So we can put a fluorescently labeled molecule, a fluorescently labeled sugar on top of these cells and look for how quickly that goes through. Obviously, the faster something makes it through, the worse the barrier is there. But we can functionally assess these cells in culture dishes. And in a number of studies that we published with the group in 2017, we identified this growth factor, this cell goody, epithelial cell goody, this TGF-beta1 was important for disrupting the epithelium, disrupting the barrier in patients with EoE. And TGF-beta had previously been shown to be elevated in patients with EoE. So this identified now that this molecule may be a great target for looking at trying to inhibit its function to then improve the epithelial barrier in patients with EoE. And in more recent studies, we looked at this molecule HIF-1 alpha and this barrier molecule Claudin-1, as well as developing esophageal organoid systems where we were able to genetically manipulate, so turn on and off at the DNA level, genes that have been shown to be altered. Those instructions, we can now mess with those instructions, turn them on too much, turn them off when they shouldn't be, and try to better understand what the role of those genes might be in developing barrier dysfunction in esophageal epithelial cells. So I'm going to talk to you in the next couple of slides just very quickly about this study here where we've looked at this hypoxia inducible factor HIF-1 alpha. So what we showed was that in the esophagus, or at least in the presence of eosinophils that were activated, there is a competition for oxygen. So there is a hypoxic tension in the esophagus, potentially, of patients with EoE because of the increase of eosinophils. So if you just stick with me here on the y-axis, this is the amount of oxygen in the tissue or in the Petri dish. These are in the black bar, unactivated eosinophils. And what you can see is once we activate eosinophils in the red line, if you follow, there's a significant decrease and a very, very rapid decrease. So this is time in minutes. So within 5 to 10 minutes, there's a rapid depletion of the oxygen in the local environment by the eosinophils. They've now sucked it all up, used it all up, and that oxygen is now not available for other cells within their environment. So why is that important? So in our mouse model, we're able to use this red stain, this hypoxia probe stain. So this is a normal mouse esophagus. You can see a normal amount of oxygen tension or hypoxia, oxygen being used within the esophagus. But what you can see in our mouse model, there's a significant increase in the hypoxic area, the hypoxic tension. So this is likely contributed to by the competition for oxygen by the eosinophils and away from the epithelium. So we wanted to better understand what might be happening to the epithelium because of this lower number, lower amount of oxygen available, this competition that's now going on for oxygen and other nutrients within the esophagus. So what we found was the signal was decreased. We looked at the transcriptome. So remember those instructions that we talked about, they've gotten mixed up. So here's our RNA, our instructions for the pastry. And instead of the HIF-1 alpha gene being expressed both at the protein level or at the RNA level, in patients with EoE, that signal is decreased and it should not be. So something went wrong. We were also able to look down a microscope. So in the brown staining here, this is a control patient's esophagus. You can see there's lots of this brown staining. That's this normal level of HIF-1 alpha in the tissue. But in patients with EoE, you can see that there's a real depletion, a real reduction in the amount of that brown staining. So there's a real reduction in this functional molecule, this HIF-1 alpha that should be there. The signal got mixed up, it's no longer there. So in a series of studies that I won't go into today, we found that that was dysregulated and it caused a defect in a barrier molecule called Claudin-1. So this Claudin-1 molecule could no longer be expressed because of the absence of this HIF-1 alpha. So we then asked, if the instructions got mixed up, could we rearrange them? So the first thing we did to try rearrange them was we used a genetic approach. So in our mice, our EBV-EDL2, this mouse esophagitis model that I spoke to you about earlier, we made a triple transgenic mouse that essentially rearranged the signal in mice so that the signal could never be switched off, to put it in most simplistic stakes. And this led to a protection of the mice. So in comparison here in the center, looking at this mouse that is inflamed, that is modeling eosinophilic esophagitis, you can hopefully see here that I'm circling. There are a number of eosinophils collecting within the epithelium of that mouse model. But in the mouse where we rearranged the signal, we changed the genes, we rearranged the genetics, we overexpressed this HIF-1 alpha molecule that was unnecessarily switched off. What we were able to do was rearrange the tissue so it looked very, very normal. And we were able to quantify this histological activity in the epithelium and show that if we could rearrange the genetics, we could decrease the amount of inflammation and rearrangements of the tissue in this mouse model. And importantly, could we fix that barrier molecule that I mentioned? Could Claudin-1 be re-expressed now because this HIF-1 alpha molecule was re-expressed? And indeed it was. So here in comparison to the white bar, the inflamed mice, in the black bar, we have the genetically altered mice, and these genetically altered mice restored or increased the amount of this molecule, this barrier molecule Claudin-1. But this is our mice. What about our patients? Can we do this in patient specimens? Well, to start with, we work stepwise in translational research. So these are a cell line that are immortal that we're able to use in culture dishes for a very long period of time. And we rearrange the signal using a non-clinically used investigational drug that could also overexpress the signal, so fix the signal. This drug is called DMOG, and it's a prolyl hydroxylase inhibitor. So by using this DMOG on our cell line, we could look then at the expression of this Claudin-1 molecule, this barrier molecule. And indeed, if we treat our cell lines or esophageal epithelial cell lines, we can see that there is an increase here in the expression of Claudin-1 in the black bar, both at 4 hours and at 18 hours post-stimulation. But most importantly, can we do this in specimens from patients, individuals who have eosinophilic esophagitis? So to do this, we took patient specimens and we brought them back to these sterile culture laminars, where we're able to manipulate those in Petri dishes, give them drugs, give them controls, and look later on at the expression of various different molecules or of the improvement or disimprovement of the function of those cells. So here we took patient biopsies and we treated them for overnight with this drug, DMOG, and we looked at the expression of this barrier molecule. And indeed, if we use this drug DMOG to restore that signal, HIF, we're able to restore this barrier molecule in both control patients, patients who don't have EoE as a diagnosis, and patients indeed who do have an EoE as a diagnosis. So this highlights that this signal, this pathway, and this molecule Claudin-1 might be of interest for future studies and therapeutic avenues to investigate for restoring the barrier in patients with EoE. So advancing the field since 2008. So go to the literature, I was told, and make the literature. So we developed cell culture models, we've developed mouse models, And we've been very, very blessed to be able to work with a human biobank to be able to look at the translational relevance of our basic research and to try and identify a number of experimental therapeutics and lead compounds to better understand improvements for barrier and for patients with EoE. And through all of these models and all of these studies, we're able to identify a number of different molecules that are worth further study to try and understand how we can fix that leak in the pipe in patients with EoE. So it takes a village, it's never alone. And I have been incredibly blessed by some wonderful mentors, colleagues, and friends along the way. So my collaborators from Children's Hospital Colorado and the University of Colorado, Denver, as well as our collaborators at the Mayo Clinic and the University of Pennsylvania, and now Columbia University. I have returned from the US 3 years ago, to this beautiful campus in Maynooth University in Ireland and to the Kathleen Lonesdale Institute for Human Health Research. And I'm incredibly blessed to work with a wonderful team of scientists who are now advancing the field in and of their own right. Most importantly, I have been very grateful for the funding support from a number of different avenues, but particularly APFED. APFED first founded the very first experiments investigating hypoxia and HIF-1 alpha in our lab and in EoE. And I will be forever grateful for that seed, the Hope on the Horizon funding that started this whole project many moons ago. And since my return to Ireland, I'm very grateful for the support of Science Foundation Ireland to keep advancing our understanding of this disease and of novel therapeutic avenues to pursue. So with that, I thank you for your engagement, for your attention, and for your interest, and I really look forward to a great discussion.
Thank you so much. You had such an absolute wealth of information, and I'm really looking forward to diving into the questions with you. So our first question that we have here is, people are curious about your thoughts if EoE has any relation to celiac disease. This individual said her family is Irish and they seem to all have EoE or celiac. And it makes her think there's a genetic link that presents either in the throat or the small intestines. And she's just also hugely thankful to you for all your research.
Well, thank you very much. Thank you. So, it's a really interesting question, and it's been one that's been rumbling along in the background for very many years. And I don't think the field, our clinicians, we don't have an answer yet. There certainly isn't a strong relationship between the two, but it has been found in some patients that they unfortunately have both diseases together. From a genetic point of view, I'm not sure that there is a commonality. We haven't seen a common pattern in the two diseases that I'm aware of. But it is really interesting because it's definitely been observed at the clinical side of things, and I've heard my colleagues talk about that. But to date, my understanding, while I don't keep up with it as much as I probably should, I'm not sure there has been a relationship between the two defined.
Great. Next question. Have you found, is it possible that an airborne or environmental allergen can trigger the esophagus, but not test positive as IgE blood test or skin prick test?
Wow, that's a very immunologically informed question, probably better served by our allergists. I genuinely would not know the answer to that. I know that eosinophilic esophagitis can be IgE or non-IgE mediated. So I think that's definitely one for your allergist.
Makes a lot of sense. What have you found causes patients to not be responsive to treatment? You've found a lot of things that are responding.
Yeah, it absolutely is where we are looking as a field now, is try to, first of all, define patients who are non-responsive. So it's really important from a research sense, do we know that they are at the cellular level not responsive, or is it an adherence and compliance issue? Is there an issue with how the medicine is being taken? Is it being taken effectively? Do we need to improve that with patients? But when we move past the clinical interface and move into the lab, there are signatures that seem to suggest that there might be a unique genetic fingerprint of patients who don't respond. And one of the studies that I'm working on in the moment is trying to identify a molecule that might be able to tell us before time whether a patient is not going to be responsive to steroids or not. So that's part of ongoing research within the group.
Okay. Well, this leads right into another question that we had. When all treatments for EoE have failed and a patient continues to have inflammation, are esophageal stents being used?
Stents. I have not heard of this, but again, probably a question for your gastroenterologist. I do know that it's very commonplace to maybe use dilation, so the insertion of the balloon into the esophagus to spread the area in within the lumen of the esophagus and try to help some of the symptoms that patients are having. But that's obviously something that's transient, it's temporary, it's done during an endoscopic procedure where the endoscope is in place. And patients do find that that can relieve symptoms for a period of time afterwards. But something more permanent, such as stents, I have not heard about. But definitely, you know, your gastroenterologist.
Okay. Next question. How does bile reflux play into EoE?
How does bile reflux play into EoE? I'm not aware that we know anything about that. From an experimental point of view, I know that bile acids have been put on esophageal epithelial cells in culture dishes, and that those bile acids can cause those cells to become inflamed and can also cause a barrier dysfunction in some cells in some patients. But the overlap with bile acid reflux in a patient who has eosinophilic esophagitis, I'm not aware of any clinical studies to examine that in more detail.
In the electrophysiological studies you showed with TER measurements in patient subsets, were these experiments done on ALI cultures or in vivo?
So both. The one set of studies was done where the sample was taken from the esophagus. The biopsy was taken from the esophagus into the lab, was grown on a lab dish, and then was analyzed there in a machine within the lab. There are devices now that have been used in preclinical research studies, translational research studies, where you can look using a probe, touching it off the surface of the esophagus in patients. It's not very commonly used, and it's fraught with complexity in its use and application, but it is getting better. And that is something that can be done in the patient in the esophagus during the endoscopic period. And there have been a small number of studies published by European groups looking at that.
Well, we've had a number of questions come in about can we get the slides afterwards, and can we get links to some of these publications? So yes, we plan to post both recording of this webinar as well as a PDF of the slides at apfed.org/webinars. And it includes all the citations that she has at the bottom, and we can certainly work together to include links to the research as well as the slides themselves. Another question is more about considering the impaired HIF signal you found in murine EoE, have you tried treating mice with hydroxylase inhibitors to promote barrier function in mice? And was the impaired HIF function related to increased HIF degradation or defective expression?
Okay, so a couple of parts to that question. Hopefully I'll remember every part. Did we try give this drug that I used in the cells in the culture dish to the mouse? We did, but as we have learned with eosinophilic esophagitis, we need to treat the esophagus topically. So that's why steroids are given topically. We tend not to give them systemically for that and other reasons. And we find that giving the drugs or patients treatments that is topical and bathes the epithelium in that medicine, in that drug, is much more effective than giving a drug systemically. And the same goes for our mice. But unfortunately, we haven't been able to, from a pharmacological point of view, get the drug, enough of the drug to be available in the mouse epithelium. But that is absolutely my desire, is to develop a better way of bathing the mouse esophagus, much like we bathe the patient's esophagus. But that's why we did the study later on where we took the patient's biopsies into a dish and gave them the drug that way. So it was to mimic that bathing situation. And we did see that there was a restoration of HIF signaling and that barrier molecule. That was part one. Part two, why or how is the HIF-1 alpha molecule degraded or downregulated? Great question. Again, part of ongoing studies in the group. So there are multiple different ways that that can happen. And all of those ways seem to be active in research that's ongoing within the group that has found that I think the body has used multiple different ways of decreasing it. So both at that intermediate, that messenger, that RNA that I talked about, but also at the protein level. So it's targeted in multiple different ways. Hopefully that's all of your questions.
Well, we have many more questions, but you did tackle that one. You talked about at one point about the reduction in available oxygen. Could it be caused by eosinophils being significant enough to be felt by the patient? Like, could it result in shortness of breath?
Very, very good question to ask. No, because the esophagus is nothing to do with where we breathe in our oxygen, our lungs. So it's a very local effect. And we know that about EoE, that it is a disease that is restricted to the esophagus. It only affects the esophagus in patients who have it. And so these eosinophils are acting in the esophagus very locally, very much within the epithelium. And so there can be no consequences of those eosinophils in the esophagus to the patient's ability to breathe or to take on oxygen within the body.
You talked, we have another thank you for you. So thank you so much for sharing this incredible information. Have you looked at peptide therapy for healing the leaky pipe?
Peptide therapy. I'm not sure I fully understand what you mean, because to me, a peptide, as a scientist, a peptide can be anything. It's a small part of a protein. So I don't know if you're able to further elucidate as to what peptides you might be talking about. Could we use peptides to treat patients with EoE? Absolutely. But again, the big boundary I see in moving forward is going to be making sure that our drugs can get into the cells that we wish them to get into. So this topical delivery of drugs to the esophagus. So whatever peptides we might be wanting to give, we need to make sure they're getting to the cells we need them to get to. But if you want to ask me more specific, I'd be happy to.
Wonderful. What is the goal for the current research? Is it to eliminate the need for patients to have to use diet and dilation as treatment options?
I think the goal is to provide more options, and for patients to then work with their doctors to define the therapy that they feel is the best for them as individuals. Because I think every therapy will have different effects on the individual. They'll have different requirements, different delivery methods, different effects on quality of life. So for me, my goal is really to bring to the table a number of different drugs, a number of different pathways that we can affect. And as we showed in those earlier introductory slides, not every patient with EoE is the same, and they're not all at the same stage at the same time. So depending on when you are talking to your doctor, you might need a different type of therapy. So it's to try give options and to understand at a molecular level what that patient might look like when they present, and for the doctor to be able to say, this is where you are at a molecular level, and these are the options that you have at a medicine level. That would be my goal. That would be my desire.
You talked about having access to different banks of samples. How can someone with EoE send their DNA to help contribute to research?
So I would definitely refer you to Seeger's website and to APFED, but a lot of that would have to happen at your clinical center. So if you talk to your doctor, if you talk to your clinical care provider and tell them that you're interested in enrolling in research, if you want to go to the Seeger website or even to APFED, I'm sure has links to some research sites and research centers. You can definitely get involved there and get more information about what kinds of studies are enrolling at the moment and where you might have opportunities to contribute and get involved as a patient.
We have two kind of related questions here, but I'll keep them separate. Have you heard of any correlation between EoE and autoimmune conditions like alopecia? This individual had both diagnoses at the same time, and they're just trying to figure things out. Maybe there's some literature you could point them to if you're aware of any possible answers.
Absolutely. I'm terribly sorry. No, I'm not aware of any commonality between alopecia and EoE, but I will definitely look, and if I find any, I can send them back on through APFED for you.
Another question from, what are the most effective treatments in toddlers that are not just temporary solutions? Are there age-specific treatments that you're looking at with your research?
For me, no. I'm very much trying to study the disease as a whole. So patients, whether they are children or adults, this disease is the same. The genes are the same. It changes over time, as I mentioned earlier. Again, it really is something that's going to be very individual and something that you would need to talk to your clinical care provider about. And as more and more medicines come on the market and come available, there will be more options. And this is what I meant earlier about my goal is to put as many options on the table as possible, because they'll all have different conveniences and effectiveness for the patient. So if you're talking about challenges of treating a toddler with EoE, you know, there will be options there, please God, in time.
Have you identified if there's any supplements that can improve the esophageal barriment? The question here is asking specifically perhaps tailored probiotics or collagen.
Very interesting question. It's not something that I have studied to date, but it is something that I am becoming more and more aware of. I wouldn't say that I'm looking at nutritional supplements in the Petri dishes just yet, but I am trying to understand the nutritional environment. So not just oxygen around those cells, but other nutrients that are really important for cells to function. So metabolites is what these molecules are called. And I'm trying to understand the metabolic environment around the epithelium and how that changes in patients with EoE. And then can we identify factors there that may be important that we can resolve the disease with? So in that sense, we will restore what's missing, much like supplements can do.
Based on your research and the information that you've read, do you have any opinions on the effectiveness of swallowed fluid inhalers, or how PPIs fit into addressing barrier dysfunction?
Great question. So we, in a clinical sense, they've not been studied with regards to, you know, barrier in the esophagus. In an experimental sense, so in a Petri dish, if you give steroids to the epithelial cell in the context of inflammation, so you give them a cytokine, a factor that's pro-inflammatory in the esophagus, like IL-13 has been identified, and you give them steroids, you will restore the barrier dysfunction that the inflammation had imposed. Similarly with PPIs, the studies to date, I have not done any, but the studies to date have looked at more so the anti-inflammatory effects of PPIs in quietening down the inflammation or the inflammatory signals that are coming from the epithelium itself. But I'm not aware of any studies that have used PPIs to look at barrier. It's a good thought.
We have so many good questions in here. Alrighty. Have you examined other barrier molecules, for example, DSG1, in vitro after treatment with DMOG, or are the effects only seen on claudin 1?
Again, another great question. So I've not looked at DSG1 specifically. In that study, we were very much focusing on molecules that we knew were affected by this HIF-1 alpha factor, and we hadn't seen any change in DSG1 in cells where we had manipulated or switched off the HIF-1 alpha factor. So where we'd altered the signal to mimic patients with EoE by decreasing it, we only saw that the only barrier molecule that was affected was actually this CLDN1, this claudin 1 molecule. We didn't see a decrease in DSG, so we didn't try to look if it was then restored. But it is a good question to ask because maybe you can amplify it. We know that DSG isn't affected by HIF-1 alpha, but it may be affected by other things.
Nice follow-up question happens to come from a different person, but on much the same topic. Given the success with correcting the claudin 1 expression, do you think that HIF-1 alpha or claudin 1 is the only molecule that's dysregulated? Or do you think you will find even more molecules regulating barrier dysfunction that are also depressed in active EoE patients?
Absolutely, yes, the latter. So we don't propose that this is the one and only molecule, but the way we started this study was we looked at the tissue for clues, and the clues led us to these eosinophils, and it led us to this competition for oxygen, this a very active state within the tissue where cells were very active. The proliferation was ongoing in the basal cell hyperplasia. Eosinophils were coming in. We knew they could consume oxygen. So we knew there was a lot of activity, and with activity comes oxygen consumption. So we knew there should be this hypoxic signaling going on in the esophagus, and we wondered what effect that had. So that's where and how we came to HIF-1 alpha. But absolutely, we totally are looking at other pathways and processes that may be decreasing barrier function. And we're also looking at other pathways that are also responsive to hypoxia, but that are not directly regulated by hypoxia, that may be affecting barrier, and trying to come up with new pathways besides HIF-1 alpha that may be druggable, that may be targetable, that may improve esophageal barrier.
Do you have any insights or thoughts on if epithelial barrier dysfunction or eosinophilia come first?
Chicken or the egg? Absolutely, we don't know, sadly. I wish we did. There's if you think about it, are you born with EoE? Well, some patients have these GWAS studies, the DNA studies that we talked about. There are some patients who have that genetic underpinning, but it's not all patients, and not all patients have the same genetic underpinning. So we don't think it's just the genes. So you can't just be born with it. And we know with diseases like EoE, it's a mixture. It's an unfortunate constellation of factors. So it's a combination of the genetic underpinning. It's a constellation of an overactive, overzealous inflammatory immune response, and the effect of the environment on that. And those three circles bring together in the center point, unfortunately, patients with EoE. So what I do know from the studies where patients have been successfully treated, for instance, in the steroid studies where they looked at barrier function, that in those patients, there was a restoration of barrier function in the impedance, those TEOR measurements, the impedance measurements that we mentioned. So there's great hope that the barrier is probably a response to the inflammation as opposed to something you are born with. But that being said, we know that barrier dysfunction is common to patients with all of the allergies, all of the atopies. So both in atopic dermatitis, asthma, and so on, there is a barrier dysfunction. So it is possible in those individuals where that genetic underpinning sits that it is a chicken or egg, whichever way you want to look at that sentence. So I think not everybody is the same, unfortunately, but I think in some patients, it may be important therapeutically to not just quieten the immune response with anti-inflammatories such as steroids, but also to potentially repair that barrier with molecules like what we're looking at.
Do you have any insights regarding new biologics like dupilumab? Do they improve any of the endpoints in relation to any of the research you've been looking at?
You know, very interesting. I am not aware if dupilumab in the dupilumab trial or in secondary studies, if they have looked at barrier molecules in it. That would be very exciting from my perspective if it was something that they had looked at. But certainly what we know so far, it isn't something that is in the sites of the research yet.
Well, thank you so much for your time today. I also want to make sure we take some time to thank our wonderful education partners for making today's event possible. We really appreciate all of their time and their commitment to help make this an option. So thank you very much to our education partners, Abbott, AstraZeneca, to Bristol Myers Squibb, as well as to Takeda. And my screen is freezing, so let me see if I can get to the next one. Alrighty. We hope that you enjoyed today's webinar and that you will join us for future webinars as well. Our next webinar is scheduled for May 26th, and we're going to be looking at the chronic nature of EGIDS. So once again, we'll be talking about EoE, but also other conditions in the EGIDS family. And we'll be joined by Dr. Carla Davis at Baylor College of Medicine, as well as Texas Children's Hospital. If you would like to continue the conversation, we hope that you will join our online community at apfed.org/connections. It's a great place to be able to have conversations with caregivers, with other patients, and really be able to connect with each other and share your experiences. We also want to make sure that you're aware of another educational resource to continue learning more about eosinophilic-associated diseases. We have a wonderful podcast called Real Talk, Eosinophilic Disorders, and our most recent episode released last week and featured a great conversation with Dr. Ruchi Gupta, talked about the comorbidity of eosinophilic esophagitis, as well as food allergy, and also talked about her new book, Food Without Fear. Finally, thank you so much for joining us. If you have any questions, please don't hesitate to email us at mail@apfed.org, and make sure to stay tuned to apfed.org/webinars for more events in the series. Thank you again, Dr. Masterson. We really appreciate your time.
Thank you all very much. Have a great day.